Okay, excuse me. Here's the second part of the microbial growth, chapter 7. This is going to be a relatively short part, but I kind of wanted to separate the nutrition from the growth. So let's talk about the study of microbial growth. The first thing that we want to talk about is the fact that growth takes place in two levels as far as bacteria is concerned. The first level is the synthesis of cellular components and the increase in cellular size. The second phase, or the second level of growth, is by numbers of populations. Population size is how we determine cellular growth. At a certain point, all bacteria are roughly the same size, and the organisms themselves don't continue to grow in size, and they begin to grow in number. The basis of population growth is bacterial replication, which is asexual replication, which is called binary fission. Here's a great diagram of binary fission. This is a little bit different than the one that's in your book. The one that's in your book is on page, I believe it's like 189. Let me double check here. Yes, it's on page 189. And most everything is the same except for the way the chromosome looks. Basically here at stage number 1, you have a young cell. That cell has its single chromosome, and that parent cell is going to begin to divide. It's going to begin to prepare to divide, and it's going to get larger. As it gets larger, you're going to have bacterial replication beginning. You see here you have your first chromosome, and now we are beginning to duplicate that chromosome, and eventually you're going to have these chromosomes on either side, and you're going to have a septum that's going to join between the two cells here. If you look here, you'll see the beginning of what is called a transverse septum. This septum, once the chromosome grows, or once the growing is a bad term, I'm sorry, once the chromosome is replicated, you'll see that it is attached to a structure in the plasma membrane, which is the nucleoid. Remember that nucleoid space where the chromosome is. Basically, the membranes are going to begin to enlarge, and that transverse septum is going to come closer together. This septum is going to be completely synthesized here once you have complete cellular separation. You've got one chromosome on one side and one chromosome on the other. You'll see here, until that septum separates, this bacteria has two chromosomes. Okay? Once you have that septum coming together, you're going to have those cells separate, and now you have two genetically identical daughter cells. In binary fission, because this is asexual, you have no new input of genetic information, and therefore these daughter cells are genetically identical to the parent. When we talk about the rate of population, we're going to talk about it in terms of generation or doubling times. I prefer the term generation time, from one generation to the next. But essentially, this generation or doubling time is the time required for a complete cycle of binary fission. Okay, you're going from parent to daughter strands. Each new fission cycle is going to increase the population by a factor of 2. One cell will give me 2. As long as the environment is favorable, as long as there are not too many toxins, there's enough nutrient, this doubling effect is going to continue at a constant rate. The length of generation time is going to be the measure at the rate of the growth of the organism. As long as things are favorable, those generation times are going to stay relatively the same. The average generation time for most bacteria is going to be between 30 and 60 minutes. You can have some shorter generation times, 10 to 12 minutes. E. coli, for example, has an optimum generation time of about 20 minutes. So if I start a culture of bacteria at 7:30 in the morning, I can look at it at 7:30 at night, and I have a significant amount of growth in just a very short 12-hour period. If I have a generation time of 30 minutes and I'm starting at 7:30 in the morning and I look at it 7:30 at night, I've got 36 generations in that test tube. Growth during generation times during the doubling rate is exponential, meaning that I have a doubling of the population with each generation. we can actually graph bacterial growth into some very specific, if we graph it logarithmically or if we graph it arithmetically. If we graph it logarithmically, that means that the generation is doubling each time, and we're going to have this nice straight line along our x-axis on the graph. If we plot it arithmetically, we're going to have this constant curved slope where it's kind of going from the x-axis up to the y-axis, and we have this nice gentle curve. Now we can calculate the number of bacteria that we have in a particular sample at any given time. If we use the formula NF equals NI times 2 to the N, where NF is the total number that I have, that is my final number. NI is the initial or starting number, and then lowercase n denotes the generation time. So if I were to take, let's say my initial is 100 bacteria in 10 hours at 30 minutes. Okay, so I have, in 10 hours at 30 minutes, I have 20 generation times. So I would take, my N would be 20, my NI would be 100, and you would do that math and you would get the equation. I didn't do that, I just pulled those numbers out of my head. So if you want to do it and let me know what the answer is when you see me next time in class, that would be great. The population of bacteria doesn't remain constant. It can't be maintained that exponential growth. You're going to have some times where you're going to have bacteria that will be plotted on a growth curve, and you'll see that we don't have that nice straight line or that nice curve, that we have some dynamic possibilities within this growth curve. We've got places where bacteria are growing very rapidly, we've got places that are growing where bacteria are growing very slowly, and we can plot this on a growth curve. You have a method to observe this population growth where you essentially start with a small number of cells, you plot that, and then you incubate them over several hours or several days. And at intervals, you will sample this broth and you will do standard plate counts and count the number of bacteria that you have. You're going to then, once you count those, you're going to plot them, and you will be able to come up with this growth curve. Here is the logarithmic. Here is our logarithmic scale. Here is our arithmetic scale, and you can see that logarithmically we have this straight line that is basically at a 45-degree angle. Arithmetically, we're going to continue to curve and go up. Here is how you go through binary fission. We've got number of cells. This is going to be generation 0. Then we're going to have number of generations. We're going to end up with 2, then 4, then 8, then 16, 32, and so on. And you can see how that would work. Some of the stages that you see on the growth curve, as we watch this binary fission go, you'll see on that last picture that we saw that each population or each generation increased the population by 2, or it doubled the population. In the growth curve, you're going to see that we're only going to have a short portion of our growth curve where that doubling of the population occurs with each generation. There are 4 predictable phases of the growth curve. The first one is the lag phase. Then you have the log phase, the stationary phase, and then the death phase. Your book calls the log phase the exponential phase, but I feel like the log phase is a little easier to remember. So when it talks about the exponential phase in your book, it's talking about the log phase. When we're talking about the lag phase, this is a relatively flat period within the growth curve. This is a time when cells are going from an old environment to a new environment, and this is a period of adjustment. This is where cells are going to get bigger. They're going to start synthesizing products that are going to be repair structures, and so you have some cells here now that are not growing necessarily in number, but more in size. These cells are not multiplying at their maximum rate. You may have some cell division, you may have some binary fission going on, but it's going to be relatively low, and most of the time what's happening here is cells are enlarging. They're in an environment that is full of nutrition. There isn't a whole lot of toxin production, and so now we have cells that can continue to repair themselves and replicate at a lower rate. The length of the lag period is going to vary from one population to another. It depends on where they came from to the point where they are now, and so the lag phase can be as short as a couple of hours, or it can be as long as maybe 5 or 6 hours. So again, this is kind of a flat or adjustment phase in bacterial growth. Once those bacteria have adjusted to their environment, then they're going to begin this logarithmic or exponential growth. When they have growth during the log phase, this growth curve is going to increase geometrically. It's going to start that nice curve that we saw on that original slide. These cells are growing at their maximum rate of cell division. They will continue to do this as long as they have adequate nutrient, as long as toxins aren't increasing to the point where cells are starting to die. During the lag phase, growth is exponential, meaning that this population doubles with each generation. Once you begin to have nutrients decreasing and toxins increasing, growth is going to kind of level off, and bacteria are going to enter something called survival mode. This is the stationary phase of bacterial growth. The rate of cell inhibition or death is going to balance out the rate of cellular multiplication. So for every cell that replicates, you're going to have that number dying off. So things are relatively flat here. So this again is another flat phase in cellular growth. This is going to be caused by the depletion of nutrients and oxygen, as well as the increase of things like organic acids, other types of biochemical pollutants that act as toxins towards these bacteria, and the bacteria then will die. You'll have the strong ones remaining, but the weak ones are going to die off. Once you begin to have too many toxins and not enough nutrient, the cells are going to begin to die off, and you're going to see that growth curve take a downward, kind of a downward dip. Cell death is going to exceed cell growth here, and you will see that the growth curve is going to start to dip down. It may level off a bit, but then it'll continue to dip down theoretically to 0. If we look at this graph here, you'll see that the log phase here is relatively flat. There isn't a whole lot going on, but now once we reach this end of this log phase, we're going to go into this very rapid exponential growth, or that log phase of cellular growth. And then at some point, you've got a big population of bacteria with no input of nutrient, and things are going to start to flatten out in this stationary phase. Once you get in this stationary phase, you're going to see that you've got bacteria that are roughly dying at the same rate they're multiplying, so you don't see a whole lot of increase here. Eventually, nutrients are going to be less than toxin production, and a lot of bacteria will begin to die off. And this death phase, let me get my cursor back here, you'll see that this death phase begins to decline, and you may have some cells remaining viable, and it may level off, but then it's going to dip down again. This does take a long time to happen. This is not exponential like the growth phase. This is going to be a phase where cell death is kind of a gradual thing. It can take weeks, maybe even months, for a particular organism to die off. We do have some other methods for analyzing population growth. One of these is we're going to look at the results from lab number 11 that we have done already, and this one of the methods here is turbidity, and we're going to use something called the spectrophotometer. And the spectrophotometer basically gauges the amount of bacteria based on how cloudy or how turbid a particular substance is. If we look at this graph here, this graph is showing something, how much light is transmitted. What we are going to be looking at is how much light is absorbed. If you look at these two samples here, we have one that's relatively clear and one that is very cloudy. Obviously, from our eye, we can tell that one has more bacteria than the other. What we can do with the spectrophotometer is see how much of the light is interfered with, and it will tell us how much bacteria there is in a particular sample based on the amount of light that is absorbed. In this picture, it's based on the amount of light that is transmitted, but again, I'll explain this more when we get into the lab and are talking about with the tubes in hand, and you can kind of see what I'm talking about. Okay. We can also count bacteria. There are a number of ways that we can count bacteria. One of them is called the direct or total cell count. Not a lot of labs do this anymore. This is where we used to get our complete blood counts. We would have somebody that would sit there and use a very special microscope, in this case a hemocytometer or a cytometer, and they would count bacteria. Basically, you put an amount or a volume of bacteria into a groove in this slide. You'd put a cover slip that has a grid on it, and then you would count them on that grid, and each one of these grids then has a specific volume that they are counting. And so you would take that number and multiply it by your factor, and you would see how many bacteria you have in a given sample. The problem with these direct microscopic counts is you can't tell which cells are living and which cells are dead, and sometimes that's very important. You need to know which cells are which. So direct microscopic count or total cell count is not used nearly as much as it used to be. You do have some other automated counting systems. The first one is what they call a Coulter counter, where it electronically scans a culture as it passes through a tiny pipette. Again, a Coulter counter does not tell living from dead, but you have something called a flow cytometer, and that flow cytometer will measure not only numbers, but it will tell cell size, and based on cell size, it can differentiate between living and dead cells. Obviously, the dead cells are going to be much smaller than the living cells, so it can tell us which cells are living and which cells are dead. You also have something called real-time PCR, or polymerase chain reaction, and this is essentially a way that you can generate copy after copy after copy of nucleic acid. If you are looking at bacteria in a particular habitat or in a tissue sample, you can pardon me, take the nucleic acid of this bacteria and do this real-time PCR, and the more bacterial nucleic acid you have, the more bacteria you have. So if you have a large amount of nucleic acid, that tells you have a large amount of bacteria in this environment or in this tissue. And this way you don't have to isolate the culture, and we can also tell which bacteria we have based on the DNA that we have. So do we have eukaryotic DNA? Do we have prokaryotic DNA? What organism is it? And so we can do this without isolating or culturing bacteria. So again, that saves us some time if time is of the essence. Here is a picture of basically a Coulter counter or a flow cytometer. Essentially, you have your pipette here, you're dispensing your liquid. That liquid enters into this tube where the tube is only big enough to take on one bacteria at a time. So they come in single file, and they pass this electronic detector that will count each of these as they pass through here. Once they pass through here, it'll go up and it will count the number of organisms that you have in a particular sample, and you'll see that this will go up to a 10 million or 999,999,999. So again, you can count your organisms with this automatic counter here. If you have a flow cytometer, what it'll do is as it goes through this electronic detector, that electronic detector will measure the diameter of the slide, and if it's below a certain number or a certain value, it will tell you that that cell is dead and it won't count it. And so you'll either have, or you can tell it to count either living or dead, or to give you a value for both. Okay, this is the end of Chapter 7. Chapter 8 is going to be coming up here pretty quick. In Chapter 8, I want you to read, before you start listening to the PowerPoints, I want you to read sections 8.1, 8.2, and 8.3. And then I want you to read Insight 8.1 and 8.3. These are going to be the ones that are in the purple boxes. I also think I want you to read 8.4 is the Insight. Let me double check here in the book. Pay attention to the diagrams in chapter 8. They will lead you along very well through this material. This is some very weighty material that we do need to take some time. And again, I want to start taking some time at the beginning of lecture to answer any questions so that we can stay on track and you guys aren't feeling overwhelmed. Yes, I do want you to read Insight 8.4. All right. Please be sure that you write down any questions, bring any questions that you have, any study guide questions, any chapter material questions. Pardon me. And we will get them answered so that you're feeling adequate and are going to do well on the test. Chapter 8 will be coming up here pretty soon.